إن شاء الله بنلتقي في الندوة الثانية عشر بعنوان الخلايا الجذعية العضلية، الاستجابة للأنماط التدريب المختلفة ودورها في الرياضة. بيكون معنا رئيس الندوة الأستاذ الدكتور مصطفى السيد طاهر، أستاذ الإصابات الرياضية والتأهيل جامعة الإسكندرية. مقرر الندوة بن شويطة بالقاسم، محاضر بمعهد علوم وتقنيات الأنشطة البدنية والرياضية جامعة قسنطينة 2 الجزائر. المتحدث الدكتور صوفي جوانيس جامعة مانشستر المملكة المتحدة. وبنسعد بوجود الترجمة دكتورة ريان جمعون، طالبة تخصص لغة إنجليزية، طالبة ماجستير تخصص لغة إنجليزية جامعة محمد نمين دباغين سطيف 2 الجزائر. دكتورة آمنة مرقسي، طالبة دكتوراه قسم علوم وتقنيات الأنشطة البدنية جامعة محمد نمين سطيف 2 الجزائر. اتفضل دكتور مصطفى. دكتور مصطفى. احمد معاك دكتور مصطفى. بنعيد على حضراتكم تاني عنوان الندوة الخلايا الجذعية العضلية.
أنا معاك يا مغازي، معاك يا أبو.
اتفضل دكتور مصطفى، اتفضل حضرتك.
بسم الله الرحمن الرحيم. بيشرفنا اليوم الدكتور بن شويطة بالقاسم، محاضر بمعهد علوم وتقنيات الأنشطة البدنية والرياضية جامعة قسنطينة 2 الجزائر. كما يشرفنا الدكتورة صوفي جوانيس جامعة مانشستر المملكة المتحدة لإلقاء محاضرة بعنوان الخلايا الجذعية العضلية والاستجابة لأنماط التدريب المختلفة ودورها في الرياضة. يتفضل الأستاذ الدكتور بن شويطة بالقاسم مقرر الندوة بتقديم الدكتورة صوفي. أهلا وسهلا.
بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف المرسلين. نشكر رئيس الندوة الأستاذ الدكتور مصطفى السيد طاهر على إدارته لهذه الندوة. أولا وقبل كل شيء نود أن نشكر كل القائمين على هذا المؤتمر الدولي المتميز في مجال الرياضة والصحة، بداية بجامعة الإسكندرية وجامعة قسنطينة 2 والأكاديمية البريطانية لعلوم التكنولوجيا الرياضة. كما نرحب بالدكتورة صوفي جوانيس من جامعة مانشستر المهتمة بالدراسات المرتبطة بفيزيولوجيا التمارين الرياضية. فأهلا وسهلا بكم جميعا معنا في فعاليات اليوم الثالث من المؤتمر الدولي. نحيل الكلمة الآن إلى أساتذة الترجمة.
Good morning, Dr. Sophie Joanise. My name is Joanne Ryan, and I will be your translator. We welcome you to start your symposium. إذا ننتظر فقط بضع دقائق لكي تلتحق الدكتورة Sophie Joanise.
Great. Good morning, everyone. Sorry, I wasn't able to unmute myself there. So I will just share my screen and hopefully get started. All right, so hopefully screen is shared and everything is okay.
Yes, everything is clear.
Okay, perfect. Thank you. So before I get started, just thank you very much for allowing me to speak a little bit about my research and research interests today. So I appreciate you guys taking the time and listening to me this morning. So what I'd like to talk about today is the regulation of muscle stem cells, which are commonly referred to as satellite cells, following different forms of training. And, you know, if these cells potentially have a role in sport. So I just kind of break down my presentation a bit today. So just the outline. So I've kind of split this up into 4 different parts. So part 1, take a microscopic look, I guess, at skeletal muscle. In the second part, we'll talk a little bit about the role of these satellite cells in muscle regeneration, repair, and remodeling. Then look at the impact of different types of exercises on the satellite cell response. And finally, wrap up with kind of a short discussion surrounding the potential role of satellite cells in sport and performance. So I'll get straight into the introduction. So kind of just taking I guess a microscopic look at the skeletal muscle. So here's a schematic representation basically of your skeletal muscle. We know that you know, your skeletal muscle is attached to the bone via the tendon here. You then see your muscle belly, so the bigger part of the muscle. And eventually, if you kind of follow this through, you get to the individual muscle fibers. Now, as a sports scientist, understanding how skeletal muscle works is a really important question because, you know, at the end of the day, it plays a really extremely important role in movement. And if you take a step back and actually kind of consider whole body movement, it really is at the basis of, you know, sport and performance. Now, I'm sure it comes as to no surprise, but, you know, skeletal muscle can adapt to a lot of different exercises. Professor Hans has just covered some of that previously, but we know that, you know, things like resistance exercise, aerobic exercise are all different types of training that people complete. And based on, you know, the sport that you're completing or the event that you're training for, you know, the training that you complete will be specific to that type of sport. So obviously, you know, this is something that's really important to really maximize training and muscle adaptation. So if we look at this a little bit more closely, if we think of, you know, resistance training, so you have this kind of image on the side here, we know that that's often associated with, you know, increases in muscle strength. So sort of a whole muscle or a whole body measure, if you will. But if you look at this a little bit more closely, more at kind of the cellular level, we know that that's associated with things like increases in muscle protein synthesis. And with this increase in protein synthesis, you can see an increase in muscle size, but, you know, that's actually also visible at the really kind of muscle fiber level, so at the cell. Now, if you think of aerobic training or endurance type training, you know, a measure that's often associated with that is increases in VO2 peak or VO2 max, oxygen consumption, during exercise. Now, although this is, again, more of a kind of whole body measure, we know that specific things are happening at the skeletal muscle level. And that could be things like increases in muscle oxidative capacity, and then also associations with sort of an increase in capillary density or the microvasculature within the skeletal muscle. So, you know, although skeletal muscle can adapt to loads of different training and a lot of different stimuli and exercise, and we can definitely measure this at the whole body level or at the whole muscle level, I think it really is important to start understanding what's happening kind of at the cellular level to enhance or to drive these types of adaptation. So if we kind of return to this kind of schematic representation of muscle, we know that skeletal muscle is one of the largest organs in the human body. And muscle, importantly, is very kind of highly metabolically active tissue. So it serves as a really important source of glucose disposal and of protein storage. And so that's at the end, at the end of the day, it plays a really important role in maintaining whole body homeostasis. Now, if we look at this a little bit more closely, we know that skeletal muscle is composed of kind of many individual multinucleated fibers. So that's sort of depicted here. So you have a lot of different individual fibers that compose your skeletal muscle. And we know that skeletal muscle is composed of different types of these fibers. So for example, you've got type 1 fibers here, which are kind of more associated with a more oxidative type phenotype, whereas you have type 2 muscle fibers, which are a little bit bigger and are more glycolytic in nature. But at the end of the day, skeletal muscle is composed of all of these fibers, and it's the ability of these fibers to work together or to work synergistically that really allows the skeletal muscle to produce force and to produce movement. Now, what's also important is in skeletal muscle is that there are a lot of different organelles that are within the muscle fibers that really support its proper function. So this kind of cartoon here, we've got mitochondria down here. So obviously we know the mitochondria are really important because they really provide the skeletal muscle with the energy to be able to complete some of these kind of movements, and gives it energy to complete or be able to produce force. Now, something else that I had mentioned that's also important is this idea of capillary or the microvasculature. So again, kind of a representation of skeletal muscle, but you see kind of capillaries dotted throughout the muscle. And that's important because, the structure basically allows the muscle to be exposed to the systemic environment. So it brings oxygen, it brings nutrients, it brings growth factors. So there's a lot of things that go into, maintaining skeletal muscle and allowing it to respond to different things. Now, one thing that's also important to consider is that although skeletal muscle is multinucleated, so each of these cells have a lot of different nuclei, which are kind of depicted here by these blue dots, it is postmitotic. So these nuclei no longer undergo cell division. However, skeletal muscle can, adapt. We know that it adapts, it responds to a lot of different things. And the reason it's able to do this is because it's home to kind of a specific subset of cells. And these are termed muscle stem cells or satellite cells, which are kind of depicted here along the side of the fiber. And it's for this reason, or with these cells, is the reason that the muscle can continue, or one of the reasons muscle can continue to adapt to different stimuli. So that's kind of it for part one.
Excuse me, Dr. Sophie Joanise, I would like to translate to the audience. Thank you. تناولت الدكتورة صوفي جوانيس في مداخلتها موضوعاً مهم حول الخلايا الجذعية العضلية. وكما هو متعارف عليه، فإن العضلات الهيكلية هي أكبر عضو في جسم الإنسان، وهي أيضاً نسيج شديد النشاط الأيضي، حيث تلعب دوراً أساسياً في الحفاظ على توازن الجسم. وهي أيضاً نسيج قابل للتكيف بدرجة كبيرة، ويستجيب لمحفزات مختلفة كالأشكال المختلفة للتمرين. تتكون العضلات الهيكلية من العديد من الألياف متعددة النوع القادرة على العمل بشكل تآزري لإنتاج القوة، ومع ذلك فهي قادرة على إعادة تشكيل وصيانة وإصلاح نفسها طوال العمر. يعتقد أن قدرتها على الاستجابة للمنبهات والتكيف معها تتم بواسطة مجموعة من الخلايا الجذعية الخاصة بالعضلات الهيكلية. You may proceed, Doctor.
Thank you. So, kind of summing up the introduction, we'll now move on to satellite cells, or these muscle stem cells, and what their role is in muscle regeneration, repair, and remodeling, really. So again, this is just another schematic representation of, or a very basic one of what skeletal muscle may look like. So here you have your muscle fiber, and as I mentioned, there's a lot of nuclei within a muscle cell. But here you kind of have these satellite cells depicted on the periphery of the muscle fiber. Now this is another image of a satellite cell. And specifically, these cells are located, again, right at the periphery of the muscle fiber. So between the plasma membrane, so the membrane of the fiber, and the basal lamina. And basically, they actually got their name as a satellite cell because they actually orbit the muscle fiber. So this is where these cells are located, sort of in a normal, healthy adult tissue. Now, these cells a lot of researchers became quite interested in them and what they're doing. Because they are in skeletal muscle, we know the skeletal muscle adapts to a lot of things. So they really became an interesting topic to study. And this was very popular in the late 1990s, early 2000s. So this is some of the first studies where researchers really wanted to look at or determine, what satellite cells look like in human skeletal muscle. So this is one of the first examples where they actually took images of skeletal muscle biopsies and looked at the satellite cell content within human skeletal muscle. Now this has evolved a little bit. So this is kind of a follow-up to some of these first images. So becoming a little bit more clear, now individuals and researchers aren't only kind of interested in describing what satellite cells look like or what their numbers are in human skeletal muscle. They like to understand the response a little bit better. So to build on that even further, we're now able to look at, so again, this is an image of skeletal muscle biopsy where we've identified satellite cells and also different fiber types. And we can even look at these cells, so these satellite cells, in relation to different structures within the skeletal muscle now. So our ability to kind of describe what's happening within the muscle, and specifically with these cells, has really evolved and allowed us to answer a lot of different questions, especially when it comes to satellite cells and how they relate to exercise and exercise training. But just a little bit more on satellite cells activation and what does it look like? What does it mean? So like I said, we know that satellite cells reside on the periphery of the muscle fiber, but they're able to become activated based on a lot of different things. So this could be anything from an actual quite severe injury to the skeletal muscle, but they can also become activated after, for example, just weight-bearing physical activity. So when a satellite cell becomes activated, it will go on and proliferate. So it'll increase its number, and we can actually quantify this within muscle. We can determine how much the number of satellite cells have increased. Now from this, a proportion of these satellite cells will also revert to quiescence. So they essentially kind of return to their starting spot on that periphery of the muscle fiber, and they really maintain the number of muscle stem cells within the muscle to allow the muscle to then be able to respond to things kind of throughout life. And then some of these cells, rather than reverting to quiescence, will actually go through and start to differentiate. So these cells, what I mean by differentiating is that they will eventually kind of fuse with your existing skeletal muscle, donate their nuclei there, and then really kind of drive or assist, you could say, or contribute to muscle adaptation or muscle remodeling. So this is a very kind of basic overview as to how muscle stem cells can become incorporated into the skeletal muscle and really drive and contribute to muscle adaptation. So one thing that, scientists and researchers have really kind of agreed upon, which is good, is that satellite cells are absolutely necessary for skeletal muscle regeneration to occur. Now, this has been shown by many kind of different groups, often using mouse models or rodent models. So this is just an example of one of the studies that have looked at this by Charlotte Peterson's group. And again, kind of using quite complex transgenic mouse models, what they were able to show is that when you eliminate satellite cells or you deplete an animal of satellite cells and you introduce injury. So if you injure a mouse that essentially no longer has satellite cells, and if you injure one that still has a satellite cell, still healthy skeletal muscle, the response is quite different. So in a mouse with satellite cells, the muscle is able to completely regenerate. So it's able to really reestablish its architecture to what it was pre-injury or at baseline. However, in the mice that don't have satellite cells, that regeneration really can't occur properly. And it really does compromise the ability of the muscle to regenerate and it essentially just never regenerates. So with this, and with these studies using kind of animal models, it really showed that satellite cells are important and are completely necessary for regeneration to occur. Which then brings us, which, you know, it's interesting in animal studies, but why do we want to study this? So as a sports scientist, why am I interested in studying satellite cells? So kind of common dogma, what's long kind of been associated between satellite cells is in humans, they're associated with skeletal muscle hypertrophy, so an increase in size. So again, several research groups have showed this, but I think this study by Verdijk and colleagues kind of really showed this nicely. So following a type of resistance exercise training, they were able to determine the change and increase in individual muscle fiber size. So they saw an increase in the cross-sectional area or in the size of individual muscle fibers. And they also determined the change in the number of satellite cells per muscle fiber. And essentially what they saw is that individuals that had the greatest change in muscle fiber size also had the greatest increase in satellite cell content. So really suggesting that the increase in satellite cells are important to support muscle hypertrophy or muscle growth in human skeletal muscle. Now, more recently, we've also been interested in starting to explore this kind of idea in non-hypertrophic type exercise, so in aerobic training or endurance training. Now, another aspect I think is very important is that in humans, satellite cells, we've shown are important in muscle injury. So when a muscle is damaged, for example, through eccentric contractions, we observe a very kind of robust expansion of the satellite cell pool. So with muscle damage in humans, you do see an increase in the number of those satellite cells. And now at the end of the day, this can have a really significant, or can have significant implications, especially in clinical or compromised populations. So for example, we spoke a little bit about aging, where, you know, it's a population you lose muscle mass. This is important to start to understand some of these things, but it also has a very kind of important role in potentially athletic populations when you start to incorporate kind of training and how that might relate to performance.
Excuse me, doctor, I'm going to translate to the participants. وفي محور الساتلايت سيلز أين يوجد هذا النوع من الخلايا بين الصفيحة القاعدية وغند الليف العضلي، حيث تلعب هذه الخلايا دورا هام في تكوين خلايا العضلات وتعويض التالف منها، كما أنها مرتبطة بزيادة حجم العضلات، ولهذا السبب كان الباحثين مهتمين بها منذ فترة طويلة، بالتكيف العضلي وكيف يمكن لذلك أن يؤثر على الأداء في المدى الطويل. شكرا. You may proceed, doctor.
Thank you. So hopefully I've kind of given you a good overview of satellite cells and how they work in skeletal muscle. So now I'd like to tackle kind of the question of what's their role in exercise and how do they react to different types of exercise training, for example. So again, this is something that many research groups have done, but I'm just showing an example here of one of the studies that I was involved in. So with resistance training, we know that a skeletal muscle grows. So this is what we've shown here. So with 16 weeks of resistance training in young males, we saw an increase in muscle size, and that was measured by an increase in lean body mass with DEXA. Also, we saw the same idea in measuring quadriceps volume via MRI. So at the whole muscle level here, but we also did see increases at the kind of the cellular level. So we saw an increase in the size of type 1 and those 2 muscle fibers. Now with this, this type of training, we saw the increase in muscle size. We did see an increase in the number of satellite cells that were also associated with type 1 and type 2 muscle fibers. So the thought is, or it's kind of hypothesized then, that to support the increase in muscle size, you need a contribution of these satellite cells to be able to sort of donate their nuclei to really allow the muscle to kind of maximize its adaptation. And with resistance training, that's in terms of growth. But like I had said, we're also interested in looking at this in terms of different types of training. So not necessarily just resistance training, but what happens to satellite cells following, for example, aerobic or endurance training. So aerobic training isn't necessarily associated with hypertrophy. So you wouldn't assume an increase in fiber size always happens. And with this type of training, what we see is that there isn't, you don't always see an increase in the number of satellite cells. So in this study specifically, we had kind of individuals complete different types of aerobic training. So more of traditional kind of continuous training. And then they also completed different protocols of sprint interval training. So with this, like I said, we didn't see any increases in fiber size, but we also didn't see an increase in the number of satellite cells with this type of training. But what was interesting is, even though we didn't see an increase in the number of satellite cells, this type of aerobic training did result in an increase in the number of active cells. So the number didn't necessarily go up, but the cells had become more active, were differentiating. So again, suggesting that, you know, although the adaptation that's occurring in the muscle is different to that we see with resistance training, these cells are still doing something. They're still kind of contributing potentially to some of that remodeling. So I think this kind of figure here sums it up quite nicely. So we know that, like I said, resistance training is often associated with an increase in muscle fiber size, and that's specifically with type 2 fibers. So for example, you have your muscle here, you undergo resistance training, you'll see an increase in the number, you'll see an increase in the size of the muscle, but you'll also see an increase in the number of nuclei within the muscle and in the number of satellite cells, suggesting that, you know, those cells are probably doing something to help mediate that. And then if you undergo aerobic training, so instead of resistance training, you kind of go down this aerobic training route, and you see not necessarily an increase in the size of muscle, but some sort of remodeling. So a shift maybe towards a more oxidative phenotype, maybe an increase in type 1 fibers. And you don't necessarily see an increase in satellite cells, but you might see more of these cells becoming activated, again, suggesting that they're probably supporting some sort of remodeling that's happening in this tissue. So again, all of these things are important when you start to try to link kind of different training adaptations and how we can really try to maximize a satellite cell response to support some of these adaptations that we need to see. So we know that satellite cells are necessary for regeneration to occur. They're also very important in adaptation to different types of exercise training. But another aspect, I guess, of training that's important is this idea of muscle repair and recovery. And I'll show you now that satellite cells are also kind of involved in this repair process in human skeletal muscle. So like I mentioned, satellite cells are able to become activated following different types of damage. So for example, eccentric contraction causes damage to some extent within the skeletal muscle. And this is exactly what we've kind of seen here. So in this study, individuals completed, or they got a muscle biopsy, completed a bout of maximal eccentric contractions. And then...
Excuse me, Professor. I'm going to... Thank you. وفي نفس المحور تطرقت الدكتورة إلى كيفية استجابة الساتلايت سيلز لأنواع مختلفة من التمرين، حيث ثبت علمياً أن تدريب المقاومة ينتج عنه زيادة في حجم العضلات، والتي تكون نتيجة زيادة هذا النوع من الخلايا. وقد أظهرت الدراسات أن التدريب الهوائي ينتج عنه زيادة في هذه الخلايا التي تساهم على ما يبدو في إحداث تغيرات في النمط الظاهري لألياف العضلات. You may proceed, Doctor. Thank you.
Thank you. So as I was saying, it just following these types of eccentric contractions where skeletal muscle damage sort of occurs, and we actually count the number of satellite cells within the muscle, what we see is that fairly quickly after this type of exercise, we see kind of quite a robust increase in the number of these cells. So suggesting that, again, you know, they're playing an important role in muscle repair. Now, what's also important is that these cells also become activated not only after kind of very heavy eccentric exercise, which is something that not, you know, every person or even athlete would incorporate in a regular type of training block, but they also do become activated after heavy resistance exercise, which is what we show here. So after a bout of heavy resistance exercise, we also did see an increase in the number of satellite cells after that, again suggesting that even though damage may not, or muscle damage may not have been kind of as widespread, it may have been much more localized, we did see an increase in satellite cells, suggesting that they're, again, contributing to muscle repair to a certain extent. So again, I think incorporated one of these schematic kind of diagrams, and I think it summarizes this quite nicely. So kind of our research group and others have fairly consistently shown that after a bout of resistance exercise or eccentric contractions, satellite cells become activated. And you can measure this kind of up to 96 hours following this bout of exercise, depending on what type of exercise was completed. Now, when you try to kind of translate this from the acute phase to what happens over training, so if you complete some of these resistance exercise kind of training bouts over and over, you would expect to see an increase in muscle size. And essentially what we see here is that eventually these cells, they become activated, proliferate, eventually will fuse to the skeletal muscle, donating their nuclei and really supporting the increase in fiber size. And just to quickly kind of go over this study, this is kind of depicting what I was describing in this schematic. So in this study, individuals again trained for that 16 weeks of resistance exercise, which did cause an increase in muscle size. And what we measured here was the change in quadricep volume, which is on the x-axis here via MRI. And then we also looked at the acute satellite cell response. So what happens to the number of satellite cells following that first bout of resistance exercise before training started? And what's interesting is that the acute change in satellite cell content was associated to the overall change in muscle volume or muscle size that we saw with training. So suggesting maybe that what happens acutely is related to what happens over kind of a training program, which again is, is quite interesting and probably has some implications when we start to consider kind of athletic populations and how to maximize a training program. So just basically kind of wrap up the presentation with this final sort of section where try to discuss a little bit on the potential role then for satellite cells and how we can kind of incorporate this or consider this when we think about sport and performance. So we know that satellite cells now play an important role in muscle repair. And I think something that's important to consider is then if the satellite cell response can be optimized, can then the repair process be accelerated? And if that's the case, can we really maximize what an athlete's kind of training program looks like, for example? But then that brings the question of how can we actually maximize the satellite cell response, and can it actually be optimized? So there is some evidence suggesting that we can manipulate sort of this acute satellite cell response. One of the ways is, well, we've shown that architectural differences within the muscle fiber can have an impact on the response of satellite cells to muscle damage. And there's some interesting work that's starting to look at nutritional interventions and how that can impact the satellite cell response. So the first thing I'll just kind of quickly go over is this idea of the importance of the microvasculature, so of kind of capillaries within the skeletal muscle and how that can affect satellite cell function or activation. So in this study, kind of university-age men, so young men were recruited, and we determined an index of skeletal muscle perfusion. So perfusion indicating kind of a high kind of capillary density. So the group was either kind of divided with high perfusion of the skeletal muscle or low perfusion of skeletal muscle. And what we saw, thought was quite interesting, is that in the group that had a high perfusion, we saw that the force recovery after damaging exercise was actually accelerated. So suggesting that, they're able to kind of gain back some of that force more quickly than in the group that had a low skeletal muscle perfusion. And importantly, this was also associated with kind of a greater satellite cell response and also an accelerated one. So suggesting now that potentially if we're able to maximize the microvasculature within the skeletal muscle, we may be able to really maximize and accelerate muscle recovery following these types of damaging exercise. So I'll also just kind of quickly go over some of the evidence that looked at different nutritional interventions and how they might affect satellite cell content. So in this first study, sort of here on the left, they completed an acute eccentric exercise bout. So this was work done from a group out of Copenhagen. So individuals completed 150 maximal eccentric contractions, so to cause skeletal muscle damage. Now, one of the groups consumed a placebo drink after that, and then one of the group consumed a drink containing both whey and carbohydrates. And essentially what they saw was that in the group having consumed the whey and carbohydrate drink, satellite cell activation seemed to be accelerated and greater compared to the placebo group that hadn't consumed that drink. So this is what happened kind of acutely, which is interesting and, suggests that maybe we'll be able to kind of further manipulate the satellite cell response. But this is also sort of looked at in kind of a training paradigm. So this is a study done by Mike Roberts' group at the University of Auburn in America. And essentially what they did was had individuals train over 12 weeks. So a resistance training type program, and they were supplemented with different types of protein beverages kind of throughout. So leucine, whey protein, and soy protein. And essentially what they saw is that each group had an increase in size. However, the groups having consumed this whey protein had a greater increase in the number of satellite cells pre to post training. So again, not necessarily sure of the exact kind of mechanisms as to why it's happening, but I think it's interesting that some of the work here is suggesting that some nutritional interventions can be applied to really maximize satellite cell function, which can potentially directly impact maybe training paradigms and performance. So one kind of final thing I just wanted to quickly touch on. So this is some work where researchers were interested in looking at non-steroidal anti-inflammatory drugs, or NSAIDs, on the satellite cell response, which I think it can be interesting, especially when we're dealing sort of with an athletic population, which they may use this kind of more frequently and throughout training. So a lot of this work was completed by Abigail Mackey at the University of Copenhagen. But in this study here, they had individuals consume NSAIDs for 2 weeks, kind of leading up to this damaging bout of electrically stimulated eccentric contractions. And what was interesting here is that when individuals consumed NSAIDs in the form of ibuprofen, they actually saw an increase in satellite cell content following this damage compared to the placebo group. So again, indicating that, maybe the consumption of NSAIDs can potentially heighten or accelerate sort of the recovery process following damage in humans. However, though, this is kind of contradictory to some other findings where following maximal eccentric contraction, again, so inducing muscle damage, where this NSAIDs were actually infused within the skeletal muscle, they essentially saw the opposite. So there was no increase in satellite cell content. So again, although the findings are kind of equivocal here, it's like contradictory, I think it is interesting just to know that, some pharmaceutical interventions may also have an impact on satellite cell kind of activation following damage. So all kind of things to consider when we're trying to potentially maximize the satellite cell response. So just to kind of quickly kind of sum everything up. So how the data that I've presented, how can that potentially support performance? So I think if the satellite cell response to exercise is optimized, the question to ask then is, can muscle adaptation then be maximized? And if the acute satellite cell response is optimized, can we maybe accelerate the muscle repair process? And now finally, if repair and recovery is accelerated, can then training programs be further optimized with the hopes of really maximizing kind of athletic performance? So I think these are all things that are important to consider. And I think future work, may be focused on kind of this last point here. So just to conclude, hopefully I kind of discussed and shown you that satellite cells are necessary for muscle regeneration to occur. And in humans, they've long been associated with hypertrophy, but they do also play a role in non-hypertrophic remodeling, shown through adaptation to aerobic or endurance exercise training. Finally, satellite cells become activated after resistance and especially eccentric kind of damaging exercise, and they do contribute to muscle repair. And then finally, there's some evidence now showing that satellite cell activation, so that acute activation, can actually be altered. So I think future work should aim to understand how to maximize skeletal muscle repair recovery, and potentially how satellite cells may play a role in this. So just very quickly, a lot of the work that I presented here was completed through my PhD and postdoctoral work over at McMaster University and Professor Johnny Parisi's lab. So just to acknowledge him and a lot of his lab group that were involved in a lot of these studies. And that is all for me.
Thank you, Doctor. We would like to thank you for granting us some of your precious time and for the enlightening knowledge you have shared with us today. وفي المحور الأخير تحدثت الدكتورة أن هذه الخلايا تلعب دورا مهم في إصلاح العضلات، فمن المنطقي أن استجابتها للتمارين قد يسرع عملية الإصلاح ويزيد من التكيف مع التدريب للرياضيين. وفي الأخير شددت الدكتورة بضرورة البحث في التدخلات العلمية التي يمكن أن تزيد من تعافي العضلات الهيكلية من خلال تنشيط الساتلايت سيلز ومن خلال مجموعة أو المجموعات الرياضية التي قد يكون لها تأثيراً كبيراً جداً. وشكراً.